It's a more open panel discussion session. I'm Elizabeth Eisenhauer, and I'm very pleased to be able to moderate this panel today. Hopefully it won't come to the needing emergency first aid equipment or anything like that. We've heard an awful lot this morning about how cancer begins, what goes wrong with us to make cancer cells grow and thrive, and how new therapeutics that might affect what has gone wrong have to be tested in people, since sadly curing a mouse isn't very predictive of being able to cure a person. So we've also heard from Dr. Barry about what happens after clinical trials are completed, and there is a belief that the results of those trials should lead to making new therapies available to patients. What's the process of evaluating the evidence and the value and so on? So this session's entitled The Pitfalls and Promise of Cancer Clinical Trials. And I'm hoping you're going to come out with a kind of a balanced view of, yes, these are really important tools to understand how we improve cancer outcomes, but there are also some challenges with how they're designed, how they're interpreted, and also how at the end of the day, the new treatments are priced for the market into which they're going to be sold. So with me today, we have Janet Dancey remotely in spirit and in physically, no, remotely physically, but not in spirit, shall we say. We have Dr. Bishal Gyawali, who you've met, Dr. Joe Pater, who's the inaugural director of the Canadian Cancer Trials Group, and Dr. Chris Booth, who's an oncologist who is interested in the value of healthcare and also global oncology. So I have a series of questions I'm going to pose to each panel member to pull out some of these ideas and themes, and there should be ample time at the end to have your questions added to the mix. So I'm going to start with Bishal. Oh, and by the way, people had the option to either present a few slides for their question or just speak to you directly. And we have a 50-50 mix of that particular thing. Good trial. So Bishal, I'm going to ask you to discuss some of the ways through the choice of how a trial is designed or the endpoints that are used to measure treatment effect, some clinical trials can have the odds stacked in favor of finding benefit of one arm over the other. Over to you.
Thank you very much, Elizabeth. First of all, it's a big pleasure to be here today, being moderated by you and being alongside Dr. Pater and Dr. Booth. So to answer your question, I have brought up a couple of slides to give a very brief overview of what things can go wrong. It does not mean that it will always go wrong. It does not mean that clinical trials are always bad. But what I'm trying to convey the message here is how we can improve the clinical trials and how we can be aware of some of the pitfalls in the interpretation of clinical trials. So a couple of points to highlight, but I'll not be covering all of them. I'll be covering the first three and the fifth one. First of all, the trials may not be measuring what matters to us. And the eligibility criteria of the trial may not be representative of the normal patient population. The control arm in the trial may not be the standard of care. And the trial might show results that are statistically significant, but may not be meaningful for the patients. So I think Dr. Booth is going to talk about this in detail later, but the problem with some of our clinical trials nowadays is that they do not measure whether patients live longer or better as a result of our drug. They simply measure whether the tumor grows from that size to this size after getting the treatment, or how long does it take for the tumor to grow from that size to this size. Does it actually matter to patients? There is evidence to show that this duration of growth does not correlate with how long a patient lives or how better quality of life a patient has. So in the absence of those benefits, does it actually matter what size the tumor is? The other problem is that of patient population. I'll give an example from liver cancer. In liver cancer, this was one of the first trials of a targeted drug where they measured survival with the drug versus placebo. And as you can see here, the average survival time, the median time was almost 11 months with the drug and 8 months with the placebo. Patients who were getting only placebo, no any treatment, were living for 8 months. But what happens in the real world when they measured? In the real world, patients who got the drug were living for an average of only 5 months. That means patients who got placebo in the trial were living far longer than patients who got the drug in the real world. Why that happens? Probably the trials are including patients who are much fitter and having better life expectancy and better otherwise than patients in the real world. The third is the control arm. In the randomized trial, they will compare the experimental arm with the standard of care. But sometimes the control arm may not be the standard of care. They might be using a treatment that is already proven inferior in the previous trials. They might not be allowing an effective drug by saying the control arm can be anything of treatment choice, but they would limit the choices to drug X, Y, and Z, and then using a placebo or ineffective drug instead of an effective or active comparator. And there is the issue of statistical significance versus clinical meaningfulness. In trial, what we look for is whether the p-value is less than 0.05. What that means is whether the results that are seen in the trial are likely due to the effect of the drug or from chance. However, we have seen several examples where the benefit is very small. In this trial, 6.2 versus 5.9 months. That's just 0.3 months or 10 days of survival benefit, but the p-value is less than 0.05. So they can claim statistical significance, but is it clinically meaningful? Another example, PFS is just the delay in the growth of the tumor size by 0.1 month, that is 3 days, but it is, the p-value is significant. Are these meaningful for the patient? That's a different story. When I'm saying that, patients might think, or the public might think, oh, so randomized trials are bad, so we should get rid of them. No. That would be just like getting rid of the word coronavirus and thinking that COVID has disappeared. The solution to these problems is to do a better randomized trial or a well-done randomized trial, not no randomized trial. And why did this happen? Why are we having all these problems? Because there is the financial interest of the sponsor to skew the results towards the drug showing benefit. And then there is the role of experts, media, and the sponsor of the trials. And there is a big role of education. I think a lot of these problems boil down to lack of education among the stakeholders. So this was a recent paper that Dr. Booth, who is on the panel today, and I wrote together, in which we asked for a common sense revolution in oncology, where we try to put patients' benefit at the center of everything, so that whatever matters to the patients should be what we are measuring in the trials. Thank you.
So thank you, Bishal. So what Bishal tried to highlight, I think, was that you can design in features in a trial through the selection of patients or what you use as a control arm, or even how you potentially analyze it to make results appear positive. And often that one-line summary at the end of the day, this was a clinically significant, or this was a statistically significant result, is the only thing that gets repeated. So it's really important to know more than just one value, or the top line summary, I guess, would be the one of the take-home messages. I think we're going to have a bunch of questions and comments at the end. Did you have something you wanted to have clarified now? And did you want to press the microphone button in front of you?
Oh, okay. So my question is, because we do these all these trials and get the data and then do the analysis, right? Everything is based on statistics. How can a trial that's statistically significant be done in such a way that it's also clinically meaningful? Like, what are some ideas you have to improve that process?
So I think we'll come back to that at the end. I mean, just as you can design in potential advantages for the outcome you want, you can design them out by asking for things that are done in the right population, that are done with a difference that is meaningful clinically and so on. But I think we should come back to that, because the next question is also about an aspect of clinical trials that, as you heard from Dr. Berry this morning, is not always available when it should be, when tough decisions are being made, and that is the impact on quality of life. So I'm going to ask Joe to speak a bit about this. a lot is written about quality of life measurements in cancer trials. And the Canadian Cancer Trials Groups was one of the first in the whole world to start measuring quality of life as part of trials. But what I want Joe to try to address is what actually does it mean? What does quality of life mean? And how did we get involved in this in Canada? And how should we be measuring and interpreting quality of life?
Okay, yeah, that's all. Thanks, Elizabeth. It's a big question, and I'll try to address it as fast as Bishal addressed his. We got involved in measuring quality of life a long time ago, actually almost 40 years, 1983, when we were doing a study of chemotherapy and advanced lung cancer. And as you've heard today, the issues are, does a treatment help people live longer, and or does it help them live better? And at the time, there was controversy about whether chemotherapy did any of that, and concern because the chemotherapy was so hard to take that even if it did prolong life, it might impair quality of life. This is surprisingly, considering it seems like we're just talking about this. There was a lot of interest in measuring quality of life back then, and I knew some of the people who were involved. And people were trying to develop ways of eliciting from patients in a way that we could measure in a clinical trial how good or bad their quality of life was. And that essentially ended up being what we call instruments, or basically questionnaires that had on them a number of questions that related to things that patients said they valued with respect to the quality of life. And these were generally things like how well they were able to get around, what kind of social support they had, what their emotional health was, etc. Whether or not these things really measure this entity, which we call quality of life, is kind of hard to know for sure. But it does measure, the instruments do measure things that do matter to patients. So we incorporated 2 of those measures in our initial study. And unfortunately, we didn't implement ways of making sure that we got the quality of life data back. And in fact, at the end of the study, we didn't have enough information to draw any conclusions about quality of life on that study. So that led us to think we need to take a more systematic approach to collecting data on quality of life. We consulted widely. We had leadership from a man named David Asoba, who was very interested in this. And we implemented a more systematic approach to ensuring that we got quality of life data on our studies. Subsequently, we actually showed, we were probably the first academic cooperative group to show that quality of life could be measured and with high response rates. And this led to implementing a policy that essentially all our trials, all our larger phase 3 trials, would have a quality of life endpoint. We learned quite a bit from doing that. We learned that giving that same chemotherapy to patients after their tumor was resected, that also prolonged life. Giving chemotherapy to people after surgery does have a negative effect on quality of life, but people recovered from that and regained their quality of life at the end of the study. And there were a number of other studies where it was an important endpoint. And so this is carried on. We're still very active. Members of our group are active nationally and internationally in collecting these data. But I think we're at a point where there are challenges, and you've heard about one of the major challenges, which is absence of data on quality of life. Even when you intend to study it in a clinical trial, even if you intend to collect data, sometimes you don't get quality of life information, either because patients get tired of filling out forms, or somebody forgets to ask, or more importantly, it's sometimes much more difficult to get quality of life information at times in a clinical trial where things are not going well, like when the disease progresses. And I think Dr. Dancey mentions we almost never try to carry on collecting quality of life data after this treatment we're studying fails for a variety of reasons, including the fact that it's That's a burden for patients that we don't want to impose on them. So, but we have an issue that we don't always have quality of life like when we would like to get it. We're studying that, trying to find other ways to improve how we can collect data when we need it. And one of the things that I think will make a difference is the collection of quality of life and other data remotely. There's no real reason why a patient has to come to a cancer center and fill out a paper form to tell us how they're doing. And we and everyone else is working on ways to collect such information remotely. And also, of course, the clinical data that goes along with it. So there are challenges, and I think you've already heard where the gaps are, and hopefully we can fill in the gaps because we do need this information to make sensible decisions.
So thank you, Joe. And I think, I mean, Joe has tons of experience with quality of life. One of the things that he taught me is that measuring quality of life in a trial that doesn't have a comparator arm sometimes can mislead you because, of course, in normal circumstances, people have ups and downs in how tired they are, whether they have an appetite or whether they are emotionally upset. So that just looking at one arm of a trial or a non-randomized trial, the quality of life, doesn't really tell you what would have happened anyway. And so that's why it's important. But the other thing he taught me is there's two important places for quality of life. One is when people really have good quality of life. They're in theory cured. You might be treating them to prevent a future recurrence, but if they weren't on treatment, they'd be feeling pretty fine. And so are you making their experience worse? And the other is at the other end of the story, when really death is in the cards, they're not feeling well, and you're attempting to palliate them. And palliation, by its very name, means symptom-related improvements. So you need to measure that if you are doing a palliative treatment. So something to think about. And the next question is to Chris Booth. And we also touched a bit on this morning. It's about the concept of value. What does value mean in clinical trials? Putting together all the elements of the trials, together with the cost of new drugs, which we also had emphasized this morning. I'm directing to you this question. Can you describe how value, maybe a concept, to pull all of that together and give an example of how assessing value can help decision makers and maybe even patients?
Great. Thanks, Elizabeth, and thanks everyone for the opportunity to be here today. So yeah, I have a few slides just to guide some comments about the concept of value, which pertains at the design phase of a clinical trial. And then it's probably most important after we know the results of the clinical trial, and we're considering making decisions with this data, either at the bedside as a doctor-patient decision, or a societal level, at the health system level, where society can pay for this. And then how we allocate different priorities throughout the journey of patients. So just a few comments. Many of our cancer treatments do provide large benefits for patients. They help people live longer lives, they increase the cure rate of cancer, and they can also improve quality of life. And so that's one bucket of treatments. Another bucket of treatments are treatments that have, moderate benefits. They help patients, certainly, but they're not home runs. The problem is this third bucket is becoming very, very common. And many of our new medicines have very, very small benefits. I'm not saying all the new medicines are small benefits. There's been some transformative drugs and major home runs that have really improved things for patients. But in aggregate, a lot of our new drugs are pretty marginal. And this is something that you don't really read about in the newspaper. It's a bit of an uncomfortable topic to discuss, but I think it's something that's increasingly recognized as what we, some of us call the value crisis in cancer. And so some of these challenges that I think were touched on this morning in the sessions before lunch, as well as comments made by Bishal, is that, sometimes we're measuring things that may or may not matter to patients. I think it's pretty clear that living longer and living better would clearly matter to patients. The extent to which some of these tumor measurements measure to patients, I think, is open for debate. The idea of an effect size, which is how big or small is the benefit? Does it help you live for an extra 10 years or 10 months or 10 weeks or 10 days? We've actually seen all of those in clinical trials. And ironically, the results of each of those 4 different trials would also land on the front page of a newspaper, game-changing drug, life-changing drug. So there's not a lot of nuance in the discussion within the public or even within our own field about how transformative some medicines are and how marginal some other medicines are. And then the other issue, which is the staggering price of new cancer medicines. So this is how I can think about the concept of value. It's a relationship between the magnitude of benefit. So how much does a treatment help patients? And then what are the costs? And the costs can be at the patient level, the health system level, or the societal level. So the questions I ask myself when I'm thinking about, reading a trial, designing a trial, or at the bedside, making a treatment recommendation to a patient, the first question is, what is the endpoint? So what improved when this medicine was given? Was it overall survival or something else? How big was the difference? And then do both of these things matter to patients? So this is a slide that shows, the cost conundrum we have in cancer medicine. So if you look at the red line, this is going back over 40 years. The red line is median household income in the United States adjusted for inflation over time. You can see it's fairly static over time. But look at the blue line. It's this skyrocketing cost of anti-cancer medicines. And we now live in an era where the typical cancer medicine costs $150,000, $200,000, $300,000 per year. And so those are very big numbers. They're big numbers for any health system. And those numbers become even more complicated when you think that, well, some of those medicines cure people or help them live for 20, 30 extra years. And some of them might not even help people live longer at all. And so these are, big numbers that need to be thought about and talked about. The other problem, and I think this was probably touched on earlier before lunch, is that unlike any other economic system that we live in, there is no relationship between how well the cancer medicine works and its price. This doesn't hold true in any other part of our life. If you want a nicer bicycle, you spend more money. If you want a larger house, you spend more money, you get a larger house. When it comes to cancer medicines, if anything, there's an inverse relationship. The drugs with the smallest benefits have the largest price tag. And that just boggles my mind, even as someone who you don't need an MBA to know that's a bit of a strange scenario. So Bishal's highlighted this. And so this is this endpoint that now has become the king of oncology, progression-free survival. And there are certain circumstances actually where it does make sense to use this. There's a limited number of circumstances where it's been proven with statistical tests that the amount of growth on a CAT scan is highly correlated with how long someone lives. Unfortunately, though, that's only a limited number of circumstances. And despite that, this is now the most common endpoint in clinical trials. And most new drugs you read about in the newspapers and that are given every day in cancer centers, they improve this. They slow down the amount of time between when that cancer on the left grows to the cancer on the right. And so you can imagine, a person, 5.5 feet, 150 pounds, and they've got a tumor that goes from this big to that big. And, you don't need to, have a medical degree to know that, it's probably not going to feel any different for the patient, and they may or may not live any longer. But this actually drives everything that we do. So this is a slide just showing kind of some of these trends. So you can see there's 3 curves here. So the blue curve shows that when Dr. Pater and Dr. Eisenhauer were young whippersnippers, running clinical trials, we were pretty smart. They were pretty smart. I wasn't around yet. But look, the overall survival is what was, they were trying to design trials to show that we could help people live longer lives. And then when Bishal and I came along, these new young whippersnippers the last 10 or 20 years, look what's happening. Overall survival has decreased over time as the main endpoint of trials, and this tumor measurement has gone up. There's a number of reasons for that. And again, sometimes it's appropriate, but most of the time, we should be very wary of that. The other curve is the amount of clinical trials funded by the pharmaceutical industry. And we now live in an era where more than 90% of cancer drug trials are funded by the pharmaceutical industry. And I'm not suggesting that should be 0. Many of our best and most important clinical trials and treatments come from excellent studies done in partnership with the pharmaceutical industry, but that pendulum has swung too far. And now it's very difficult in the current ecosystem to fund other questions and trials that matter to patients' lifestyle, reducing intensity of treatment, improving side effect profiles, et cetera. So this is just, I guess, kind of where we're at now. So the majority of new cancer medicines that you read about that are approved and that are put in guidelines, they actually don't help people live longer. There's no evidence that they do that. It's usually these tumor measurements, but some of them do improve survival. But even when you look at the ones that help people live longer, again, there's some home runs there that are transformative, but in aggregate, it helps people live for an extra 3 months. And I don't want to take away 3 months from anyone, but we have to remember that those 3 months come at a cost, and not just a societal cost. So there's clinical toxicity with side effects for patients. There's financial toxicity with costs associated with care for the patient, travel, missed work, out-of-pocket expenditures. Needless to say, there's huge system-level financial costs. There's an emerging concept that we've been talking about more lately called the time toxicity, which is the amount of time that a patient invests in seeking cancer care. Days traveling, days spent in the chemotherapy unit, blood tests, biopsies, CAT scans. By definition, these are days that cannot be spent with family, traveling, visiting with grandkids, or pursuing other interests. So there's a time cost there. And then this concept that Dr. Eisenhauer mentioned before, which is what happens when these medicines are used in the real world. And what we've shown with our group, and other groups have shown the same thing, is that these benefits that we see in clinical trials, they're always smaller in the real world. And that's because patients in the real world are older and sicker and have more complicated cancer. So if we have a medicine that helps someone live an extra 8 weeks in the clinical trial, it's almost certainly less in the real world. Is it 6 weeks, 4 weeks, 3 weeks, 2 weeks, sometimes maybe 0? So these are some of the challenges that I think we need to reconcile. And again, when we criticize some of these problems, it's not to say that we don't need clinical trials to answer questions. In fact, that would be the biggest mistake we could make, is abandoning clinical trials. It just means that we have to be maybe a little bit more thoughtful and think about these issues when we design the trials, and also as policymakers, patients, and clinicians, how to interpret those results and frame them with patients.
Great. Thank you very much, Chris. And I think we're going to hear later from Judy, because one way, of course, to make clinical trials as meaningful as possible is to have patients engaged in the questions that are asked, the differences that are looked for, and the potential impact on quality of life. So I think you also set us up nicely for Janet. I can see you over my shoulder, but you probably can't see anything about me, maybe the mask. Because as you touched on, a lot of this meeting has been really around new treatments, new drugs, pharma industry, cost. But that doesn't mean that that's the only thing clinical trials look at. So this question for Janet, can you discuss what types of other questions besides a new drug type question are important, and the role that academic clinical trials groups like the Canadian Cancer Trials groups have or will play in addressing those questions? And I look forward to what you have to say.
Thank you, Elizabeth. And hello, everybody. I'm going to try to keep my comments brief because I'm really interested in the questions and hearing the audience comments. So I'll make 4 points. So first of all, I think we all agree that cancer care is more than about drugs. And one of the key things that academic clinical trial groups and clinical trialists bring is an interest in studying cancer patients and the issues of cancer patients and cancer treatments in a more holistic and broader fashion. So we're the groups who are more likely to design trials around radiation therapy or surgery therapy, around supportive care measures to improve the symptoms that cancer patients may be experiencing, and testing lifestyle interventions or psychosocial interventions that may improve the outcomes and the well-being of cancer patients. The second thing that academic clinical trialists are interested in and have had a leading role that Joe has touched on has been around methods improvement and endpoints improvements. So a lot of the work that we do is really about how can we improve clinical trial designs and how can we improve clinical trial endpoints. And in addition to the seminal work and the development work around patient-reported outcomes and health-related quality of life, In addition, Elizabeth led the original effort to standardize the imaging-based endpoints of progression and regression that are currently widely used, although those were initially developed to standardize the assessment of anti-cancer effects in early clinical trials. And that methods development is ongoing, because as you've heard, the issues with trial designs and endpoints are ongoing. The third thing that academic clinical trialists bring is a willingness to share data from the clinical trials. And the trials that we do, we are very interested to aggregate those data with other groups who've done similar trials, because there is a value in terms of getting a bigger data set across multiple trials of patients who've received similar treatments in similar situations, because that bigger data set allows us to look for effects in subgroups of patients, in older patients, in patients with certain comorbidities or on other drug treatments. And the fourth thing I think that we bring is a perspective around economics and quality. So our group and others were interested in designing trials. We don't have the funds and the resources of a pharmaceutical company, and we don't have that profit motive around bringing a drug to market. So our trials tend to be focused on endpoints and effects that align with what patients, clinicians, and policymakers are looking for in terms of magnitude of effect, magnitude of benefit. And in addition, we tend to run our trials in the most cost-effective manner that we can, focusing on collecting data that's really quite important and ensuring high-quality data collection for those data elements. So on balance, I would say academic groups bring a perspective and reflect a perspective that is more holistically focused on cancer patients and the benefits that can be derived from treatments for them. And we're the groups who are most likely to design our trials around questions that are most important to Canadians and relevant to the Canadian healthcare system. And at the end of the day, I think we bring a perspective that can marry what is of value and what is important to policymakers and payers, to patients, to our clinical community, as well as our research community.
Thank you, Janet. I think you've heard a number of different ideas and perspectives, and at this time, happy to take questions. And maybe we'll start with the question that we heard right at the very beginning after Bishal, which is, given that sometimes, as you point out, statistically significant trial results are not clinically significant, how would you change the design of trials differently? And it isn't just for you, it's for anyone, to say that what we are looking for are clinically meaningful differences in outcome.
Yeah, I think this will also tie in with the discussion about quality of life. We had recently written a paper where we argue that in quality of life literature, we see that they predefine the threshold for what to call meaningful quality of life benefit. But we don't see that as explicitly stated for efficacy trials. So that means even before starting the trial, there should be clearly laid out what magnitude of benefit would be considered clinically meaningful. We know that a p-value of less than 0.05 would be considered statistically significant. But what is the minimum magnitude of benefit that we're expecting to see in the trial? Is it a 3 months difference? Is it a 2 months difference? Is it a 4 months difference? If we explicitly lay that out, then that will also help us with planning the sample size and the power calculation, but it will also inform the public and the potential participants of the trial that, okay, this is what we're expecting to see in the trial. This is what we are going to test. And that way the patient can also make an informed decision about getting enrolled in the trial.
So Joe?
This is a vexing problem. And I think I read one of the papers I read on this said it's an insolvable problem. The difficulty we face in doing a research project like a clinical trial is that we're going to do one experiment and we're not going to get to do 300 experiments. So the we have to accept that the result we get, it could be, even though what we postulated when we started the trial was true, there will be random variation about what we postulated. So if the classic framework for designing clinical trials, the Neyman Pearson approach, where we take into account power and statistical significance, we hedge our bets. We say, okay, we think that a treatment should prolong survival or double median survival, or any way we want to express that. And so if we base the calculation of the study so that if that's really true, we have a pretty good chance of having a statistically significant result. But if you kind of play that one out, you will realize that because there is random variation among in the results, we can't stop, we can't avoid the possibility that even if what we postulated is true, we might get a result that is not as extreme as that, but is still statistically significant. The only way to avoid that in entirety is to do a trial with a 50-50 chance of getting a positive result, which we generally don't want to do. And so I think what Bishal and Chris have suggested is a useful idea that we should define in advance what we think is would be a meaningful result. And when we do our sample size calculation, see what the chance of what we look at the smallest result that could be statistically significant. And if that result is way below what's meaningful, that we have to rethink how we calculate the sample size.
Yeah, just to echo that, I mean, Joe's a lot smarter than me, so he can think about this more deeply. But my simple answer is we design smaller trials to avoid the risk of finding these small benefits. And so if you have a smaller size, you won't have the statistical power to find a difference. It's 10 days, 20 days with a p-value in general.
But small trials are less reliable.
Correct, but with a larger sample size, I agree. But for example, we've done a modeling exercise where if you have funding and enough patients to run a single trial of 1,200 patients for a cancer where they're going to live about a year, you can power it to show an improvement in, I can't remember exactly the numbers, but let's say 2 months of improvement with precision. You could actually run 3 clinical trials with 400 patients each to answer 3 different questions, and you won't have the power to look for a 2-month difference. You'll have a power to look for 3, 4, 5 months difference. And so you can answer 3 questions. Now there's a chance that you're going to miss out on a difference at 6 weeks, but you can also ask 3 questions, identify larger effect sizes. But it's not quite as simple as that, but that's a general idea.
So Janet, did you want to weigh in?
Picking up on a couple of points, I agree that a key way to improve the situation is if there was a coming together of the relevant stakeholders and interested parties, which includes patients, policymakers, regulators, industry, and researchers and clinicians around what is clinically meaningful differences to help us design the trials. Because the trial designers, whether they're academic or industry, always start from that position anyway. But that perception of what is meaningful actually is different depending on which individuals you're talking to at any given time. There have been steps in that direction, of course, where professional organizations who are not industry-led, such as the American Society of Clinical Oncology or the European Society, who have put together consensus frameworks around what is a clinically meaningful difference in certain settings, curative settings and metastatic settings. And I think they're good benchmarks. As Joe said, however, we design our trials to try to ensure that we're not going to miss an effect due to chance alone if there is a real effect of that size there. And so sometimes if we end up, despite designing the trial to show and detect a clinically meaningful difference, the results of that trial are a statistically significant difference that may show a smaller effect. And that's just the elements of the design. Now, the interpretation of that result and the willingness to pay the price of the treatment is actually something that should occur subsequently. The other area I think we really need to focus on is improving our endpoints and our presentation of the results on those endpoints for people. Because we tend to try to reduce the complexity to very simple things, and that does a disservice. So we've touched on the issues of endpoints, measuring quality of life, measuring progression, which Bishal and Chris highlighted, but highlighted from the perspective of how a measured difference may not show clinical benefit, but not necessarily how a difference associated with progression really could be clinically meaningful. We also keep reducing differences in survival to medians. And yet one of the most influential papers I've ever read was by Dr. Gould, and I recommend it to everybody. The median is not the message. You have to look at the totality of the evidence to really make an informed judgment about the value, I would say, of a treatment.
Great. We have time for one more.
Yeah, so just again, from the standpoint of cancer patients, and I think patients' involvement in these studies, and my knowledge of an interest in the whole biotech sector, this question of quality of life. I mean, if you're a biotech healthcare company, the costs with developing this stuff is just staggering, right? 90 right. How many drugs never get to market because very expensive industry. But has anybody talked to the drug companies with this from the standpoint of this quality of life question? In terms of how they might change the trials, is that something that has been discussed? Because to me, what is important to me these days is my quality of life. And
So I'll just quickly say many drug companies include quality of life measures in their trials. Some don't. And many drug companies that are hoping to get a new drug on the market discuss the design with the FDA or with Health Canada or the EMEA, the European agency, beforehand, and would receive advice to include it if it is especially in a more of a palliative setting. That being said, sometimes those data are not made as readily available when they report the results of the trial. So I think that's where pressure from patients and the public is going to be really helpful. Because if they have the data, they should share it. It should be part of the publication. It should weigh into the discussion of the pros and cons of treatment. So, I mean, the example that Scott was talking about this morning, the key clinical trials didn't seem to have much in the quality of life space. And then there was some real world quality of life data, but it isn't quite the same as knowing in the clinical trial whether there was a decrement in quality of life that needs to be taken into account in the decision making to take the treatment. Because any real world collection of data, like a survey, you're going to be picking the people who want to reply because they did well. So it's very biased source of data. It really needs to be part of the trial. And many trials include it, not all present it. And sometimes they present it in a way that sounds positive, but is really just simply not negative. So as informed members of the public, asking for really high quality clinical trial data on the symptoms that matter most to patients especially is really key.
so we can't force them to do that, or they're not required to do that.
There's no requirement as far as I know, Joe, or no. There would be, Janet's going to answer that. There's certainly guidance from the regulators to include it.
So the requirement to include it depends on what the clinical trialists, or in this case, industry, may want to claim as benefit should the trial be successful. So what if, for example, the treatment claim might focus on patients having improved cancer-related symptoms, pain, etc. Then yes, there's a requirement to include an instrument and a measurement and an assessment and then an analysis and present that to demonstrate that, that improvement on a patient-reported outcome. The challenge, I think, for quality of life is around what specific aspect or what specific tool is being used to make claims about effects on quality of life. And it's actually more precise to focus on measuring changes in cancer symptoms, either improvements of symptoms at baseline or delay in the development of cancer symptoms. Similarly, it's easier to measure specific impacts of certain treatment-related toxicities. The hard part is actually measuring what we really are interested in measuring, which is the global effect on patients and patient well-being. That actually is not necessarily sensitive to differences in treatments in the clinical trials, because the differences in treatments being tested in a clinical trial are probably less in terms of their impact on quality of life than all of the other things that are impacting quality of life of patients with cancer. So it really depends on what is the likely claimed clinical benefit or the endpoint that will lead to regulatory approval about whether these endpoints are going to be included in clinical trials.